Welcome to BioBytes with Mick. I'm Mick, and in today's video I want to start building a foundation. It's easy to talk about the high-level cool biotechnology like tools and uses and applications and how AI is shaping most of those changes. And don't worry, more videos on this another time. But in order to discuss some of these high-level concepts and these newer concepts, I want to build a bit of a foundation or some reference towards some of the most basic tools and concepts used in the biotechnology field. So to start us off, let's discuss a common tool used in genetic engineering, PCR. PCR, or Polymerase Chain reaction, is used, talked about, and literally sung about quite often, partially because, at least in my opinion, it's a very simple and easy to understand concept. PCR is a technique that allows us to replicate DNA fragments in only 3 steps. And by repeating these steps over and over again, we can make more and more and more copies of our selected DNA fragment. The goal of step 1, melt, is to turn dsDNA, or double-stranded DNA, into ssDNA, single-stranded DNA. How do we do this? Well, we crank up the heat. Raising the temperature of the solution to 95 degrees Celsius is just enough to separate the hydrogen bonds connecting the complementary A to T and G to C base pairs. Any more than that, and we're looking close to 100 degrees Celsius, which, you might remember, is the boiling point of water. Step 2 is anneal. In this stage, DNA primers bind to the ssDNA strand and flank the amplicon, the region of the gene that we want replicated. In the final stage of PCR, extend, a DNA polymerase binds to the primers established in step 2 and moves alongside the ssDNA strand, adding on complementary nucleotides as it goes. Often, a DNA polymerase from Taq, a heat-loving bacterium, is used. The Taq polymerase can sustain the high temperatures and can polymerize about 1,000 nucleotides a minute. So we know what PCR is, but what is the point of PCR? Why do we need more DNA? Well, essentially, it comes down to making something that's super small, undetectable, to something more visible and usable. PCR amplifies the target DNA to a level where it can be seen, measured, and/or manipulated. For example, in forensics, we can use PCR to identify suspects from trace genetic materials left on the scene. PCR helps us in genetic testing to help identify specific genes or mutations associated with diseases. Once the DNA is amplified, it can then be used in DNA sequencing, gel electrophoresis, techniques that I'll discuss in future videos. We'll wrap up today's video by discussing the 2 different kinds of PCR. Traditional, or endpoint PCR, versus real-time PCR, also known as quantitative, or Q, or RT PCR. A traditional PCR is more or less what we've been discussing this whole time. Melt, anneal, extend, repeat, and do that as many times until you've amplified your DNA region to an extent you think measurable enough. Then we can do other tests to measure the amount of DNA. Real-time PCR is the same in that the same steps, melt, anneal, extend, are used. What's different is the amount of dsDNA present after each round can be reported and quantified by the amount of fluorescence the sample is giving off at the time of measuring. So essentially, fluorescent dye is added to the solution, binds to dsDNA strands, and as more DNA accumulates, the more there is in the sample, the more fluorescent you have, and there's a machine that can read the amount of fluorescence the sample is giving off. Keep in mind, this is just a basic introductory video into what PCR is and how it's used. If you're interested in learning more about PCR, do your research on things like optimal annealing temperatures, visualizing PCR results on gels, and the problems or troubleshooting often involved in analyzing these results. That, in a nutshell, and a small one at that, to be honest, is PCR. If you've liked this video or just learned something new, make sure you subscribe and hit the notification bell. I hope I'll see you in the next video as we continue our BioBytes journey.